Hello everyone, my name is Rahul. In this video, we are going to discuss about biotechnology, principles and processes. So the term biotechnology was given by a scientist named as Carl Ericke. Now, biotechnology, it deals with techniques of using live organisms or enzymes from organisms to produce products and processes which are useful to humans. Biotechnology is basically of two types. First is the old or the traditional biotechnology, which is based on natural abilities of organism to produce certain substances, such as there is production of penicillin by Penicillium notatum, making curd, bread or wine. These are all microbe mediated processes. This is also a form of biotechnology. Next is the new or modern biotechnology. This is our area of interest in this chapter in these videos. So it is based on recombinant technology, based on recombinant DNA technology. What is recombinant DNA? We will discuss later on. So, two scientists, we have to read about them first. First is Paul Berg. Paul Berg is known as the father of genetic engineering. He is known as the father of genetic engineering. Second, we have to know about two more scientists, Stanley Cohen and Herbert Boyer. Stanley Cohen and Herbert Boyer. Herbert Boyer, he was born in 1936. He was an assistant professor at University of California. So what Herbert Boyer did in 1969, the years 1969, he performed studies on restriction enzymes. He performed studies on restriction enzymes in bacteria. These enzymes had very useful properties. He observed that these enzymes had the ability of cutting DNA strands, ability to cut DNA strands. Stanley Cohen, he was a researcher, he was a Stanford scientist. He was studying plasmids, studied plasmids. Plasmids are extrachromosomal, single-stranded genetic material which can be found in certain bacterium. So he was studying plasmids, and he had developed a method of removing these plasmids from the cell and then reinserting them into another cells. So combining these two processes, we had the basis upon which biotechnology is founded. Based on these processes, the principles of biotechnology were founded. So in this video, we are going to focus more on tools of recombinant DNA technology. Now, recombinant DNA means DNA which has been manipulated to insert foreign genes. Recombinant DNA is a DNA in which foreign genes are present, which are normally not found in that particular DNA. So, in the tools of recombinant DNA technology, we will first focus on the enzymes. You all know that enzymes are basically large biomolecules. Enzymes are proteins. So, in recombinant DNA technology or in biotechnology, we use basically four types of enzymes. So, starting first with the lysing enzymes. These enzymes are used for opening up cells to extract DNA, to extract DNA or genetic material. So You know that the basic structure of a cell is such that there will be a plasma membrane, inside there will be nucleus with a nuclear membrane, and DNA or genetic material is located inside the nucleus. So in order to extract this genetic material, we have to do away with this plasma membrane. We have to break the nuclear membrane, and then only we can get the genetic material which is located inside. So in order to do that, we have enzymes called as lysing enzymes, which are used for opening up of cells to extract DNA. What they will do is they will basically digest these membranes, and we will have the genetic material which we can use for our biotechnological processes. So bacterial cells, they are usually digested with an enzyme known as lysozyme. Lysozyme is also used for human cells. Plant cells, we use cellulase and pectinase. This basically dissolves the cell wall, and fungal cells, we use chitinase. So these enzymes, they will help in opening up of cells to get DNA for genetic experiments. Next, we have a very important group of enzymes known as cleaving enzymes. Cleavage means to cut. So these enzymes are used to cut DNA molecules. They can be of two types. One is known as exonuclease, other is known as endonuclease. Exo means outer, endo means inner. So exonucleases, they will remove nucleotides. They will remove nucleotides from the ends of DNA, from the ends of DNA. So suppose if this is a DNA molecule, so what exonucleases will do, it will start digesting the DNA molecules from the ends. So it will remove the genes from the ends. So we will have like this and this. Compared to endonuclease, they make cuts at specific positions, at specific positions within the DNA, within the DNA. So if this is a DNA molecule, an endonuclease will cut from within, such that we will have fragments like this. So exonucleases, they remove nucleotides from the ends, and endonucleases, they make cuts at specific positions within the DNA. A very special type of endonucleases that we have to study is known as restriction endonucleases. Restriction endonucleases. These are also known as molecular scissors. These are also known as molecular scissors or molecular knife. They belong to endonucleases class of enzymes. Restriction enzymes, they are used in recombinant DNA technology because they can be used in vitro. In vitro means in a laboratory outside the normal body to to recognize and cleave within specific DNA sequence typically consisting of 4 to 8 nucleotides. 4 to 8 nucleotides. So these enzymes, they have very specific sites on which they are going to act. Each enzyme has got its own site and it will not act on any other site. This sequence of 4 to 8 nucleotides where an restriction enzyme works, it is known as restriction site. This site is known as restriction site, and this restriction site is palindromic sequence. It is a palindromic sequence. Palindrome means words that when read from either starting or the end, they spell the same. So within our DNA, there are certain sequences which if read in direction 5 prime to 3 prime or in the direction 3 prime to 5 prime, they are still the same. For example, we have this sequence. This sequence, when read from 3' to 5' or from 5' to 3', it will read the same. So this is an example of a palindromic sequence. Now, these restriction endonucleases are naturally found in bacteria. So what is the function of these restriction enzymes in bacteria? They bring about they bring about fragmentation they bring about fragmentation of viral DNA without affecting bacterial genome. So if there is a bacteria which is being attacked by a bacteriophage, which is basically a virus, so what these restriction enzymes will do, they will cut the viral DNA without affecting the bacterial genome. So the viral DNA will not be able to survive inside the bacteria, and hence the virus will die and the bacteria will not be affected. Now, why does the bacterial DNA itself does not get affected? Because there is a special modification present in the bacterial DNA, which is known as methylation. Due to the addition of certain methyl residues at various locations, the bacteria's own DNA is able to resist the action of these restriction enzymes. So the first restriction endonuclease, I am going to write it as RE. It was hind second. This was isolated from a bacteria known as Haemophilus influenzae. Extracted from a bacteria known as Haemophilus influenzae. So hind 2, it will recognize a specific site in the DNA, which will be a palindromic sequence, and it is going to cut at that location. Arbor, Smith, and Nathans, three scientists named Arbor, Smith, and Nathans, they discovered a restriction enzyme known as EcoR1. They discovered EcoR1, which is a restriction enzyme. Now, besides hind2 and EcoR1, we have 900 different restriction enzymes. We have around 900 different restriction enzymes which perform different function. By different function, I mean which cut the DNA at different sequences at different sites, and they are used in biotechnology. Now, how these enzymes are named, the nomenclature includes three letters. The nomenclature includes 4 letters and a Roman numeral. The first letter indicates bacterial genus. The first letter indicates bacterial genus from which the enzyme has been taken. It is written in italics. The second and third letter indicates species. The fourth letter indicates the strain, of bacterium. And a Roman numeral, it signifies the order in which the enzyme has been isolated, was isolated. So 1 means it was the first enzyme to be isolated from that bacterium. Second means it was the second enzyme. For example, we can take an example of an enzyme known as EcoR1. Now, E stands for Escherichia, CO stands for coli, so E. coli, R stands for strain, R is the strain, derived from the strain, and 1 signifies the order in which the enzyme was isolated from. So it means it was the first enzyme to be isolated from this particular strain of bacterium. Now, restriction enzymes, when they cut the DNA, they produce two types of ends on the basis of their mode of cutting. The first end is known as a sticky end, and the other end is known as a blunt end. Now, most of the enzymes that we will study or we will deal with, they produce a sticky end, and there are also many enzymes which produce a blunt end. So a sticky end, it is also known as a free end. It is also known as a free end. The free end appears something like this. The free end, it will appear something like this. So this picture shows the action of enzyme EcoR1. So this is a palindromic sequence. This sequence is a palindromic sequence. So what EcoR1 does is it cuts the DNA in between the guanine and adenosine residue. So on both the strands, it cuts between the guanine and adenosine residues. Similarly, it will cut the foreign DNA and the vector DNA. What is a vector? We will deal later on. So after cutting, it will produce these types of ends. When you will put these two strands together, these ends will automatically stick to each other. They will automatically form the hydrogen bonding and the recombinant DNA will be formed. So these type of ends are known as free ends or sticky ends. So free end or sticky end, it is produced when the cutting is a little apart from the center. So this is the center of this sequence. So the restriction enzyme is cutting a little far from the center. It is cutting here and it is cutting here, which is a little far from the center. So free ends are produced when restriction enzymes cut the strand a little away from the center, little away from the center. So this leaves single-stranded portions at the ends. These are overhanging stretches. They are called as sticky ends. They are named because they form hydrogen bonds with their complementary cut counterparts as soon as you will put them together. So for example, EcoR1, it will produce sticky ends. Hind3, it produces sticky ends. BAM-H1 and SAL1, all these enzymes, all these restriction enzymes, they produce sticky ends. The blunt ends, they are produced when the restriction enzyme will cut the DNA molecule right in the middle, right in the center. So when the cutting will be like this in the center, blunt ends will be produced. And these ends will appear something like this. So these are known as blunt ends. They are produced when both strands of DNA are cleaved in the center at the same nucleotide position, Examples: SMA1, ECO R5, Hi3, they will all produce blunt ends. Now we have other group of enzymes which are known as synthesizing enzymes. So synthesizing enzymes, they are used to synthesize new strands of DNA, synthesize new strands of DNA. These synthesizing enzymes, they are of two types: DNA polymerases and reverse transcriptases. Reverse transcriptases. The function of DNA polymerases you must have read in detail in the chapter Molecular Basis of Inheritance. So DNA polymerases, they help in synthesizing complementary DNA strands. Complementary DNA strands on on DNA templates. Reverse transcriptases, they also help in synthesizing complementary DNA strands, but on a RNA template. Next, we have joining enzymes. They help in joining DNA fragments. These are basically known as ligases. So, enzyme which is joining two DNA strands, it will be known as a DNA ligase.